Good afternoon, everybody. Today we are discussing the next topic, that is the representation of an organic molecule. In the last class, we have discussed the three-dimensional structure of an organic molecule, and today we are discussing how we will represent this three-dimensional molecule in a two-dimensional space or two-dimensional method. So there are four different methods for representing an organic molecule. First is the Fischer projection, second is the flying wedge, third is the Sawhorse, and the fourth is the Newman projection. So first of all, we have to move into the Fischer projection formula. So in an organic molecule, the central metal atom is always carbon. So in the Fischer projection, we have to represent the central metal carbon atom by a cross. And if you are observing the molecule just like this, I have shown an observer is observing the molecule. If you are observing the molecule in the same case of the observer, we can see the atom C is in the left hand side and atom D is in the right hand side. As you know that the carbon has four different valency, and here in this molecule the four valency are satisfied by one is by A and second is by B and third is by C and the last one is by D. So if you are observing the molecule, the atom C is in our left hand side and atom D is in our right hand side. So these two atom C and D is represented by a horizontal line, and the carbon, the C, atom C is in our left hand side. So that's why we have to represent the C, the atom C in the left hand side of the horizontal line, and atom D we have to represent the atom D in the right hand side of the horizontal line. And while we consider A is the top of the observer and B is the bottom of the observer. So we have to represent the atom A and B by a vertical line. In the top we have to represent A and in the bottom we have to represent B. This is the Fischer projection representation, and this is the simplest method of representing a three-dimensional molecule in the two dimension. But even then it has some of the limitations because we cannot represent a larger molecule like this, and in a most complicated molecule we have little confusion about the position of the atom. That's why we have another representation, that is the flying wedge. So here in this representation we can see that there are three different type of the line we can see here. So first is a line, the second is a solid line, and the third one is a dotted line. So the solid line, the line is representing the plane of a paper, and the solid line is represented by, I have shown that solid line in the bottom of the slide, which represent the atom which is coming out of the paper of the plane, and the dotted line is representing some of the atom which is in the out of the plane of the paper, that is in the backward side of the back side of the plane of the paper. So these are the representation. So in flying wedge we can see the three type of line. One is the line, the second is solid line, the third is the dotted line. So we are observing the molecule. We can see that the central carbon atom, and there are four valency, and the two valency are occupied in the plane of the paper, and one of the valency is satisfied by the atom which is coming outside or the top of the plane of the paper, and the last one, the last valency are satisfied by an atom which is in the bottom of the paper. See here I have shown one paper in the slide, and above this an observer is pointing a pen above the paper. So consider the 2 of the valency of the carbon atom is existing in the same paper, and the 1 is in the above the paper, that is represented by the pen, and the other is in the back side of the same paper. This is the flying wedge representation. So in this representation also there are so many limitations. That's why we have another representation. This is again the flying wedge representation. I am keeping a methane molecule in the plane of the paper. So we can see that the line CHH, which is shown by an line, is exists in the plane of the paper, and the solid line which is pointing in hydrogen atom is upward the plane of the paper, and the hydrogen which is represented by the dotted line, it is exists in the back side of the plane of the paper. Next we are moving into the another representation, that is the Newman projection. So in the Newman projection, I have shown this observer. I have shown the observer by an arrow, dotted arrow. Here we can see that consider an ethane molecule. So ethane molecule, the molecular formula is CH3, single bond CH3. So there are the two carbon atom is existing in the ethane. The first carbon atom is represented by a blue carbon, and the second carbon atom is represented by the red one. So if you are observing the molecule like this, that is in the same direction of the dotted arrow, we can see the front carbon atom, that is blue carbon atom, and we cannot see the CC sigma bond, and similarly we cannot see the red carbon atom. That's why if you are observing the molecule like this, the front carbon atom, that is blue carbon atom, in Newman projection formula is represented by a dot, and the 3 of the valency of the blue carbon atom is satisfied by the hydrogen atom, and which is also represented like this in Newman projection formula. So the hydrogen atom is represented by, in blue is the 3 valency of the front blue carbon atom, and the back carbon atom, that is carbon which is represented in the red color, we cannot see that carbon atom, but even then in Newman projection formula that carbon atom is represented by a circle, and all the 3 valency of the carbon atom is represented by the hydrogen, that also is represented by, in Newman projection formula, by a red hydrogen atom. So the figure which is shown in the right hand side is the proper Newman projection representation of an ethane molecule. So if you are observing the molecule, we cannot see the CC sigma bond directly, and we can see the front carbon atom and the other valency, and the back carbon atom is represented by the circle, and other valency of the back carbon atom is represented like this. So this is the Newman projection formula. We are usually using the Fischer representation and Newman representation in this chapter. Now we are moving into the other representation, that is the Sawhorse representation. See here, the figure which is shown in the left hand side is a Newman representation of a molecule. If you are observing in the same direction of the arrow mark, we can see, that means we have to observe the molecule in the top of the Newman projection formula. We can see the CC sigma bond. So that is represented by a CC sigma bond is represented by a line. So in the Newman projection formula, we can see the front carbon atom in a red ball, and which is represented in the top of this line, and the back carbon is represented by the green circle, and here the black carbon is represented by a green ball. And here in the Newman projection formula, we can see that the red ball is attached to the 3 of the valency. One is hydrogen, the other is OH, and the next one is CH3. So similarly in the Fischer projection formula, we can represent the hydrogen OH and CH3. See here in the Newman projection formula, OH group is in the left hand side. So similarly in the Sawhorse representation formula, OH group is in the left hand side. And hydrogen is in our right hand side. So similarly hydrogen, we have to represent the hydrogen in the right hand side of the Sawhorse representation. And methane group of the red carbon atom is in the bottom. So similarly in the Fischer projection Sawhorse projection formula, methyl group are in the bottom. So we can represent the other valency of the back carbon, that is the green carbon, in the same model. This is the Sawhorse representation formula. I hope that you understand the 4 different representation, and we are usually using the Newman projection formula and Fischer projection formula in the stereochemistry. Thank you.